I'd like to now introduce the first of today's keynote speakers, founding member Bob Weinberg. Bob has been leading the field of cancer research for over 30 years. He quite literally wrote the book on cancer biology. How many of you have seen this book, this textbook? All right, a lot of hands go up. All right, so how many of you have your signed copies? Well, some, okay, great. When this book was first published, and I'm going to see if I can embarrass Bob here, but it's actually, it's not so hard, it's not so easy, but when this book was first published, Nature, the magazine Nature said, there is no comparable text in cancer biology and no single book that is so current and informative. No, Bob, I have more to say. Right. Bob, stay in your seat. This is going to go on for another, your talk doesn't start for 10 minutes or so. Yeah. Bob also authored the single most cited article ever published by Cell magazine. It was entitled The Hallmarks of Cancer. It was first published in the year 2000. Again, it went on to become the most cited article that Cell had ever published. And so popular that in 2011, Bob and his co-author returned to update the Hallmarks of Cancer, the next generation. And actually, it was such a big deal. Cell made, Bob wants me to stop this, but anyway, Now, as you might imagine, Bob's work has led to numerous awards and recognitions, including the Gardner Foundation's International Award for Achievement in Medical Science, the Max Delbruck Medal of Science, the Killian Award, the Otto Warburg Medal, the Wolf Prize. Bob wants to enjoy this directly. The Wolf Prize in Medicine, and the National Medal of Science. So given Bob's extraordinary record of achievement, you might find it noteworthy that he would describe his current work on cancer stem cells as the most exciting of his career. And so it gives me great pleasure to welcome Bob Weinberg, who will be speaking on cancer stem cells, a new target in the fight against cancer. So.
David, thank you for that over-the-top and thoroughly embarrassing introduction. I've been here now 27 years, and I still look forward to coming to work every day. It continues to be an exciting place, and I want to emphasize something that David said, or extend it, and that is part of the reason why it is so exciting and dynamic is that we have lots of young people flowing through here all the time. Undergraduates, doctoral students, postdocs. Many research institutions are separated from universities. They don't have to do teaching, and they think that's a great thing. But I can tell you that in the great majority of cases, such institutions within a generation become rather stale and isolated and lose their former luster. And so to the extent that we have kept what we started out with, I believe much of it is due to the fact that we're deeply integrated into an academic community. Whether or not they pay our salaries, that is MIT does, is a secondary issue. What's most important is that our intellectual vibrancy comes from this unending stream of very bright young people who populate our labs and who make the discoveries. That having been said, I would like to go to my theme of today, which is the cancer stem cells, how they arise and what implications they hold for our treatment of solid tumors. And as you may know, for certain kinds of malignancies, we have had some remarkable advances over the last generation. But for other advanced malignancies, including solid tumors of the colon and the lung and the pancreas, we haven't really made many breakthroughs yet. And part of the reason that is because we didn't understand some of the fundamental organizational plans of tumors. This has really only come into light over the last 5 years, and has really revolutionized our thinking about how tumors should be treated, as I will try to indicate momentarily. So it's not as if we already knew everything we needed to know 5 or 10 years ago to develop new definitive cures for cancer. On the contrary, this field is still rapidly moving, and to my mind, it would be foolish to say that we know enough now to treat cancer the way we need to treat it. We're learning new things every month and every year, which are totally realigning our conceptual framework about how the disease arises and about how it should be treated. So just to begin, I'll show you an experimental puzzle which really represented part of the entree of my laboratory into this area. A postdoc in my lab took normal human breast tissue and he propagated in 2 different kinds of culture media and made 2 different kinds of normal human mammary cells, because there are multiple normal cells in the normal human mammary gland. And then he took these cells and he transformed them. And when we use the word transform, we mean converted them from normal cells into cancer-forming cells, thereby creating 2 different kinds of breast cancer cells constructed artificially in the lab rather than arising spontaneously in the human body. And then he took these cells and began to examine how they behave. If they were put in mice, they formed tumors, which indeed you would imagine, because the consequence of transformation is to create cancer cells. And the cells looked quite different under the microscope, as you can see, indicative of the fact that they were different kinds of human breast cancers. But what was most startling and required much subsequent investigation was the following. If we took these cells, the B type, one needed to inject 1 million of these cells into a mouse in order for a tumor to form. And conversely, the other cells, over here, the A type, I call them arbitrarily here, between 10 and 100 sufficed to generate a new tumor when injected into a mouse. And this really has some stunning implications for the way that cancer tumors are organized, because it suggests, in this case, that out of 1 million cells in this tumor here, only 1 of them is capable, upon being introduced into a mouse, of generating a new tumor. Conversely, among the population of cells forming this tumor, 10,000 of them, any one of 10,000 can do exactly the same thing. In other words, the representation of what one can call tumor-initiating cells is drastically different. And one obvious implication of that is that not all the cells within a tumor are functionally equal. Some have tumor-initiating capability, and others do not. In this case, the vast majority of the cells in this tumor cannot generate a new tumor. And this begins to take one ever more deeply into this rapidly changing field, that is the field of tumor-initiating cells. Here is one representation of this from a paper that was published from the University of Michigan in 2004, where the cells in a human breast cancer were separated by a technique called FACS that we needn't get into. Each one of these dots represents a cell from the tumor, obviously in an electronic form, an electronic representation. Here's the great majority of the cells in the tumor, and here's a small minority, and they were separated apart, one from the other. These cells, the majority cells, one needed 20,000 of them to be injected into a mouse in order for a new tumor to arise. In other words, the other 19,999 did not have tumor-initiating capability. Conversely, here, as few as 200 cells were sufficient to generate a new tumor. In other words, at least one of those 200 cells could be a tumor-initiating cell. And therefore, one began to realize that there were different subpopulations of cells within tumors that behaved differently one from the other. It was not as if we had previously assumed that all the cells in a cancer are equally capable of generating new tumors. Here was a demonstration that that was not the case. Some are far more equal at tumor initiation than are others. Of some interest is the following. If you take out these tumor-initiating cells, you can seed an entirely new tumor, and then when you extract the cells from this new tumor, you get the same pattern over again, and in fact, you can do that indefinitely. In other words, what happens here is that when this cell generates a new tumor, it once again creates a heterogeneous population of cells, some new tumor-initiating cells, and other cells which are not tumor-initiating. In other words, the tumor-initiating cells can spawn two subpopulations once again, those that are capable of generating a new tumor and those that lack that ability. And this has gone against, as I said before, our preconception about how tumors are organized. If you would have asked me 10 years ago, what about the cells in this tumor and the cells in this tumor? These, by the way, are not tumor cells at all, but connective tissue cells. If you would have asked me about them, and I would have said, if you pluck this cell out, it is equally capable of generating a new tumor as this cell, or this cell, or this cell. The same thing here. Each of these cells is biologically equal. But I've just shown you evidence that in fact they're quite unequal one from the other. Some are capable of seeding a new tumor, others lack that ability, and the tumor initiating cells that can seed a new tumor, again, are in the small minority in general. This is really much more what a tumor actually looks like. This isn't really a tumor growing in a mouse. It happens to be growing in a petri dish, but it illustrates quite graphically what I'm trying to argue. Here's a cluster of tumor cells. Here are some tumor-initiating cells. They're stained red for reasons we needn't get into. The bulk of the cells here are stained green, and they lack that tumor-initiating capability. So here you can see it directly, that there is heterogeneity within a tumor. Some cells are quite different by this level than are yet others. And so we begin to grapple with the consequences of this heterogeneity and, of course, the causes of it. Here's, for example, a case of a fatal brain tumor, glioblastoma multiforme, which continues to be a major killer. And if we look at the cells under the microscope, or we look at them in this kind of analysis, we realize there's two subpopulations. Here's the minority subpopulation. They have these dark nuclei here, and these are the cells that can initiate a new tumor. In other words, they're here in the small minority, unlike the bulk of the cells in this brain tumor, which lack the capability of initiating a tumor. Here actually is another glioblastoma visualized where the bulk of the cells in the tumor aren't even seen here. All we're looking at, there are hundreds of glioblastoma cells here. The only ones we're looking at here happen to be the rare tumor-initiating cells, which, as you can see here, interestingly, and for reasons we don't understand, happen to like to exist right next to blood vessels. That's where they home to, for reasons we don't understand. Again, I'm not showing you all the hundreds of other cells in the tumor here, which are in no way indicated in this three-dimensional reconstruction. These are all these illustrations coming, to state the obvious, from other labs. So this begins to implant in our mind the following kind of organizational scheme, which I will embrace with ever greater fervor as the morning goes on. Here we have tumor-initiating cells, and they're depicted here arbitrarily in gray. And the tumor-initiating cells can seed an entire new tumor. They can also make new tumor-initiating cells. Once again, this tumor-initiating cell can make another tumor, this can make another tumor, and in fact, within a tumor as a whole, there may be 100 times as many of the red cells as there are of the gray cells. And this scheme seems to explain much of what I've just told you. Importantly, however, it contains within it the following nugget. If we look at one of these gray cells, we realize that when this gray cell, this mother cell, divides, it creates two daughters who are unequal one with the other. It creates a red daughter and a gray daughter. The gray daughter is an exact replica of mom, and therefore, to the extent that this cell division has occurred, the number of gray cells is not depleted, it's unchanged. One pre-exists and one survives after the division. And a new red cell occurs, and we call this, by the way, asymmetrical division. When a cell divides, it doesn't make two identical daughters, it makes two quite different daughters. And this kind of thinking, this logical organization, has been studied for decades under the rubric of studying stem cells. We look at this graphically with the following two notations. Here's once again a mother. She regenerates one of her two daughters as herself, and a second daughter which becomes different. The red daughter is very different from the mother, and in fact this red daughter can begin to now become more and more different from the original stem cell up here. Another way of noting this, which I'll show with ever increasing frequency here, is the following, that when this cell, the blue cell divides, she regenerates herself and she generates a daughter. These are just two different graphical notations that essentially mean the identical thing. And so now we can begin to embrace this scheme, and introduce it into our discussions of stem cells. These days, when people think about stem cells, they think about embryonic stem cells. Indeed, the embryonic stem cells of the sort, the pluripotent stem cells that Rudolf Jenisch was just recognized for at the White House. And here are some of these induced pluripotent stem cells. These are very primitive embryonic cells, and importantly, they can do two things. So these cells are very similar to cells that are in the early embryo, and like all other stem cells, they have two traits. The first trait is they can make more copies of themselves, and I've just told you that. So they can do so indefinitely. And the second independent trait is that they can differentiate. And when I talk about differentiation in this context, I mean the fact that these cells up here have no special tissue-specific characteristics. They're very primitive cells. They're neither red cells, nor muscle cells, nor bone cells. They're this most simple embryonic cell that one can imagine, but spawned here among the descendants of these cells are a whole variety of cells which are specialized to take on many of the attributes that one needs in order to construct functional organs. So here, once again, we have these two different fates of the daughter cells of a stem cell. On the one hand, being able to make more copies of itself, and on the other hand, being able to spawn more differentiated descendants. And this is the essence of the stem cell. In this case, the embryonic stem cell. But importantly, throughout our body, there are stem cells that exist throughout our lives. It's not as if stem cells exist briefly and ephemerally during our early development and then disappear. Most of our tissues are maintained throughout adult life by small populations of stem cells which renew themselves and renew the tissue. Here's a hematopoietic stem cell, as it is called, which resides largely in the bone marrow. And this resides in the bone marrow throughout our lives. You can take this hematopoietic stem cell, take it out of the bone marrow, and inject it into the bone marrow of someone whose entire resident bone marrow has been wiped out by chemotherapy or by radiation. This would be a bone marrow transplant. And this single stem cell here can differentiate and produce all the different kinds of red blood cells, white blood cells that exist in our circulation, thereby creating the immune system, thereby creating the oxygen-carrying red blood cells and the platelets that enable clotting, and so forth. So here we have the idea that the stem cell can not only self-renew, as I show you here. Here's that reflexing arrow once again. That's its one attribute, but it can also generate all of these descendants. And there are yet other examples of what we call adult stem cells. That is, stem cells that are committed to create and maintain and orchestrate the biology of one or another specific tissue. Here's another kind of stem cell. It's called a mesenchymal stem cell, and it can renew itself once again, or alternatively, it can spawn these more specialized, differentiated daughters. In this case, those that can produce bone or fat or cartilage. And these two kinds of stem cells are what are called oligopotent. They can make different kinds of differentiated descendants, not just one kind. But whether they can make three, or in the case of the hematopoietic stem cell, more than a dozen distinct types of descendants, the overall scheme is the same. This dichotomy between self-renewal and differentiation, which these stem cells can mediate. And here's yet another. The bulk of our mitotic activity inside our belly happens every day in the lining of the intestine. And here we see it at the bottom of, for example, the colon. Outside here is the cavity of the colon. Here is a so-called deep cavity, which is called a crypt. And lying at the bottom of the colonic crypt are these stem cells, which are continuously renewing themselves and generating more differentiated descendants. And these differentiated descendants move up here, and they then become the lining of the intestine. And in so doing, they take over the work of ensuring that the wall of the colon is maintained intact and continues, in addition, to exercise its digestive functions. And we can depict this whole scheme in this very schematic fashion here. Once again, the stem cell renewing itself and generating large numbers of descendants. Of additional interest here is the following. By going through one division, this stem cell can create this daughter, which can then expand and create 100 descendant cells which have taken on the job of maintaining and orchestrating the physiological functions of the tissue. And that means that the stem cell only needs to divide once to generate this enormous horde of descendants. And that's good because that means the stem cell doesn't need to be going through frenetic, unceasing rounds of growth and division, which is dangerous because by growing through many rounds of growth and division, this stem cell might inadvertently damage its DNA. And therefore, nature has evolved things so that stem cells divide every once in a while, but nonetheless succeed in generating these enormous numbers of more differentiated, specialized progeny. In the context of cancer, we now return to the way a tumor is organized because we now realize it's organized exactly in the same way, in the same hierarchical fashion as normal tissues are organized. At the top of this, at the apex here of this hierarchy, is a self-renewing stem cell, which in this case we call a tumor-initiating cell. Again, the operational definition, because if we take that blue cell out and we put it into a mouse, a tumor will arise. Coexisting with that stem cell in the tumor are a large series of cells that have exactly the same DNA makeup, but have become more differentiated. Here is the vector of increasing differentiation. And even though these cells down here have the same genes as the stem cell, they have given up the option of ever renewing, of ever being a stem cell again. And so the bulk of the cells within a tumor lack the ability to seed a new tumor because they are no longer tumor-initiating stem cells. They're not qualified to do so, and apparently they've irrevocably moved in this direction. And I say apparently, I'll move to this theme later. And this also has an interesting implication for the process of metastasis, because we know that when a primary tumor sends off cells to distant tissues in the body, those cells must be capable of seeding at a distant site in the body. Once they land there, they must begin to colonize that distant tissue and form a whole flock of descendants, a large number of descendants, to generate a clinically detectable metastasis. A metastasis that's 1 centimeter by 1 centimeter by 1 centimeter has 1 billion cells in it. And therefore, I indicate the fact that when a cell leaves the primary tumor, if it's going to succeed in seeding a metastasis, a flock of metastatic descendants, it will need this self-renewal capability. It could be that these cells down here, one or another of these cells, will leave the primary tumor and seed a distant organ site. But it will fail in its agenda of creating a metastasis because it's lost its tumor-initiating capability. In this case, we could call it metastasis-originating capability. And so this scheme begins to inform our thinking about metastatic spread, which is not an inconsequential issue. About 90% of cancer-associated mortality is associated not with primary tumors, but with their derived distant metastases. I'm going to shift gears now, and I'm going to talk about another subject which is ostensibly totally unrelated. They'll converge later, but I'm just telling you that what I'm about to say has nothing to do with what I told you before. And this is a gear shift lever. I couldn't find anything better in Google Images. And so now we go back to biology, but a different kind of biology, the organization of epithelial tissues. This is an epithelial tissue here. Obviously, it has more than just epithelial cells in it, but it's called an epithelial tissue. The skin is an epithelial tissue. The lining of the esophagus and the stomach is an epithelial tissue, as is the lining of the lungs, the alveoli in the lungs, and the mammary ducts, and the ducts in the pancreas, and the ducts in the liver, and so forth, and even the prostate. So this overall architectural plan has been used time and time again over the last 600 million years since this organization was first developed. And what we see here, there's two kinds of cells. Epithelial cells which form the lining. Outside here is the cavity. We can imagine this is, for example, a milk duct, and out here is where the milk is being produced, or a duct in the kidneys where urine is being produced. Here's the epithelial lining, and underlying the epithelial lining, out here is the cavity. Underlying the epithelial lining is this whole assemblage of what are called mesenchymal cells, or mesenchymal, depending on what side of the pond you live on. And so here we have two different kinds of cells, and they're very different. The conventional images, ne'er the twain will ever meet. Either your epithelial or your mesenchymal, and that's not a minimal or subtle difference, because epithelial cells up here have a whole set of properties that makes them very different from mesenchymal cells. For the purpose of our discussion today, epithelial cells form these large sheets, and they don't move around very much. And if one damages them a little bit, they're very sensitive and they'll die quickly. The mesenchymal cells underlying them are moving around all the time. They're constantly reconfiguring the tissue. If you prevent the mesenchymal cells in your joints, for example, from constantly moving around, soon you're going to look like the Tin Man in The Wizard of Oz, because they're continually remodeling and reshaping the tissue. And this means that they have motility and they can actually invade. In other words, they invade through one another. They don't invade up into the epithelium, but they're constantly swarming around. Two very different kinds of cells, but of importance to us is that 80% of human cancers, the carcinomas, the so-called carcinomas, come from these kinds of cells. When these cells become converted from normal epithelial cells into tumor cells, they generate what is called a carcinoma. The mesenchymal cells are rarely involved in cancer formation. They create sarcomas, by the way. But again, these are very different kinds of cells, and that relates also to what we'll see momentarily in terms of the progression of cancer. What one has learned over the last 30 years is that sometimes epithelial cells can become converted into mesenchymal cells. And this was heresy in the minds of some cell biologists, because I told you that epithelial cells are one class and mesenchymal cells are the other class. But in fact, there is a possibility of interconversion. Here's a sea urchin embryo. This is a rather stunning scanning electron micrograph. And believe it or not, sea urchins are more closely related to us than we are related to flies and lobsters and so forth. A very important fact in one's everyday life. Anyhow, so here's an early sea urchin embryo. And what you see, the outside, this is an epithelial construction here on the outside, the earliest embryo one can imagine. These cells here have delaminated, they've removed themselves, they've become detached from the outer epithelium, they've rounded up, and now they're climbing up these fibers in the middle of the embryo. By the way, this embryo is so small you can barely see it with the naked eye. And these cells, in the case of a vertebrate, might begin to form the skin and the nervous system. These cells in here are going to form the internal organs, for example, the connective tissue and the gut. These cells have undergone an epithelial to mesenchymal conversion, because now they're moving around, they're invading into the embryo, they're no longer forming this outer sheet that I described before. Here's yet another manifestation of this. This is what a vertebrate embryo looks like very early in life. Here's the beginnings of the nervous system, the spinal cord. And these cells here, these blue cells, are emanating from the spinal cord, and they're moving, and they've started to become mesenchymal because these cells are eventually going to disperse throughout the entire body. These blue cells are the precursors of the melanocytes in our skin. They start out in our nervous system, but by the time you are born, these melanocytes have distributed themselves all over the body. To state the obvious, they give our skin its pigmentation. But in order to move from this site near the origins of the nervous system all over, they have to become mesenchymal in order to move out, because epithelial cells, as I mentioned before, are immotile. They can't move at all. Here's what we look like. It's quite a bit different from a sea urchin embryo, as I showed you before. But here we also see, this is more of a mammalian embryo. These cells up here are epithelial. These cells have become mesenchymal. They've detached from the upper epithelium, and here they've rounded up and they're actually starting to invade down here. Once again, we see these examples over and over again in terms of normal development, where epithelial cells become mesenchymal, a transformation which is called in the trade the epithelial mesenchymal transition. Here's at least 3 examples of epithelial mesenchymal transitions which play critical roles in normal embryogenesis. In fact, this EMT, this epithelial mesenchymal transition, was invented maybe between 600 and 650 million years ago. Of course, some people say, why don't you know this number more exact? And if you don't know it more exactly, then you don't know anything. But you'll forgive me that imprecision. And here we see in a whole series of different organisms where these epithelial mesenchymal transitions play absolutely critical roles in development. Here's what we look like. This is a mouse, but we and mice look pretty much the same. And we see that EMTs are also playing an important part in mammalian development. So they're not an aberration. They are, much more than we realized 10 or 20 years ago, an intrinsic, inherent part of normal embryonic development. And we now call this a program. In other words, when a cell undergoes a whole series of different changes, it really is considered a coordinated program rather than just a single change. A whole series of changes in a variety of different components. The nasty secret is that carcinoma cells that I told you before opportunistically exploit this EMT program in order to acquire many mesenchymal traits. Carcinoma cells, as I implied, originate as epithelial cells, but when they become mesenchymal, when they go through an EMT, an epithelial-mesenchymal transition, they now acquire traits associated with high-grade malignancy, and here I give you one manifestation of that. In the red are epithelial cancer cells, human breast cancer cells, and you could call them carcinoma cells because they're epithelial. The green cells are no longer red. They've in fact shut down the manifestation of an epithelial state, which in this case is indicated as redness, and instead they've become green. They've turned their back on their epithelial origins, and they've now become mesenchymal. One of these cells already here, this one here, is starting to invade into the nearby tissue. And you begin to see that this creates a cell which is biologically very different. A whole series of things change when the red cell becomes green. It's not just going from red to green, whatever that signifies at biochemical level. There are hundreds of things that change when an epithelial cell becomes mesenchymal and acquires, in this case a cancer cell, acquires all these traits. Here's yet another tumor. Here we see an EMT where here the epithelial cells, indicated here with the yellow, have become mesenchymal. They've undergone a profound shift in their biology, and these cells, I will tell you, are aggressive, whereas the epithelial cells are not. So the EMT program plays a very important role in the whole biology of cancers, because when carcinomas undergo an EMT, when carcinoma cells opportunistically upregulate this early embryonic program, cells that were previously sitting around and not moving very much, cells that were non-aggressive, by turning on this long latent program, which is hidden away in their genes, the carcinoma cells suddenly become motile and aggressive, and they can start invading and even metastasizing. What that means is that carcinoma cells are not clever enough to invent this whole program of high-grade malignancy and metastasis. Instead, carcinoma cells reach back into their origins, into the early embryonic origins, which are laid out in their genes, albeit silently. They reactivate, they resurrect this early embryonic program, and the resurrection of this early embryonic program confers in one fell swoop multiple traits of high-grade malignancy on the cells that have turned green. Here I'm only showing you one of them, the invasiveness of this cell, but I will tell you without proving it to you that this cell here is not only more invasive and aggressive, it's more resistant to different kinds of therapy, and it can move around in sites in the body where its epithelial precursors could never venture. So there are many different advantages that are conferred by cancer cells, carcinoma cells, when they opportunistically upregulate the EMT program. Some time ago, a postdoc in my lab, his name is Sendurai Mani. I first met him in Bangalore in India, the son of a rice farmer from a small poor village, came to my lab, and he was doing work on this in this general area. And he was interested in the question of what happens when an epithelial cell goes through an EMT, that is a mesenchymal cell. For him, the question was, what's the product when an epithelial cell goes through an EMT? And if you listen to everything I said now, until now, and take it seriously, which itself might be a mistake, then you would imagine that an epithelial cell makes a mesenchymal cell, and a mesenchymal cell is, by all standards, a fibroblast. Fibroblasts are all over their body. They're standard hardware, connective tissue cells, which enable many of our tissues to hold themselves together. And in fact, the answer to this question was so obvious that one might consider it inane. But fortunately, he didn't ask my opinion. He didn't ask what I thought of this question, posing this question, which is good, because I often tell people in my lab to run off on their own and not consult me, since my preconceptions may sometimes sterilize their otherwise rather substantial creativity. So he undertook a fingerprint test. Here are connective tissue cells, bona fide fibroblasts, and here are epithelial cells that have been forced through an EMT. So these are the products of the EMT I talked about before, and these are bona fide fibroblasts. And as you can see from this fingerprint test here, they look different. The products of the EMT are not standard off-the-shelf fibroblasts. They look very different by this fingerprint test, whose details needn't occupy us at present. So he concluded the following, that the product of an EMT is not a fibroblast. Of course, that raised the question, what is the product of the EMT? If an epithelial cell makes a mesenchymal cell, what kind of mesenchymal cell is it? And then he went back to his desk drawer and he looked at a microscopic section of a human mammary duct that he had done together with a surgeon with whom we work at the Brigham and Women's Hospital. Here's a cross-section of a human mammary duct. And what he saw here in regions of this mammary duct were cells that seemed to have undergone an EMT, but this was a normal mammary duct. This wasn't some kind of tumor. This wasn't a wounded tissue, this was a normal tissue, and perhaps more importantly, these cells were residing in locations where one knew stem cells could be found. So he then made the speculation that, the following speculation, a daring and a rather improbable speculation, that the product of the EMT I told you about before is not a mesenchymal cell, it's a stem cell. And this conclusion, this speculation, promptly earned him the derision and ridicule of all of his lab mates. Of course, I only heard about this later, because what if you have somebody in your lab and you're working on two different subjects? You're working on the EMT and you're working on cancer stem cells. And somebody in the lab says, well, they're really the same thing. The EMT actually creates stem cells. If you hear something like that, then almost always you think that this is the product of a fertile imagination, an overly ambitious, opportunistic postdoc who wants to describe an entire plan of all modern science by uniting everything together. And therefore one is skeptical, as were his lab mates. But he soon shut them up. Because keep in mind now, this is the question that was being posed. Is there a connection between the EMT I told you about before and the stem cells I'm telling you about now? And the way he and another postdoc worked this out is they separated non-stem cells here from stem cells up here, similar to the diagram I showed you before. So to make a long story short, if you're looking at stem cells, each one of these dots is a cell. The stem cells reside up here in this graph, and the non-stem cells reside down here. And you can separate them, obviously, and quantify them. So what they did was, these two postdocs, they took these different regulators that are normally able to program on their own EMTs, and they forced these cells to go through an EMT. In other words, they took these genes out of early embryos and now imposed them on an adult non-stem cell, and here's what they found. The cells, the non-stem cells, when they were forced to go through an EMT here or here, they migrated en masse from the non-stem cell position to the stem cell position, here and here. At the same time, by the way, the epithelial cells became mesenchymal in their shape. This is the way epithelial cell layer looked. This is the way mesenchymal cell layer looked. So this really was, at least for me, I think for them too at the moment, although they only told me about this weeks later, a stunning discovery, because it meant that the EMT actually creates stem cells. Who would have thought? Certainly I would have never guessed that. In fact, when they collaborated later on with Cornelia Polyak, who works at the Dana-Farber, she added something else to this, which is only the beginnings of a discovery. If you look at normal mammary stem cells in a normal breast, and you look at carcinoma cancer stem cells in a human breast cancer, they're very similar in their behavior, in their molecular biology. In other words, obviously cancer stem cells and normal mammary stem cells aren't the same, because normal mammary stem cells can make a normal mammary duct, cancer stem cells can make a breast cancer, but the organization that they use, the organizational machinery is very similar. They use the same stem cell program. So that represented an extension of this early thinking, and it led to a realization that maybe one needed to confront what is a major problem in current clinical oncology. Internalizing and accepting momentarily these cancer stem cells, one has confronted over the last 5 years the following issue. Let's say this is the organization of a tumor. We looked at this before, and there are many more cells here. Here are the cancer stem cells, down here are the non-stem cells. For many kinds of therapy, if you treat a breast cancer, for example, for many kinds of therapy, the tumor will shrink. But the problem is, the therapy is killing off the non-cancer stem cells, the non-stem cells within the tumor, and the stem cells, many of them, will survive. For various reasons that we can go into, the cancer stem cells in that tumor are actually more resistant to killing by many standard forms of therapy. And that creates a serious problem. If you look at the tumor as a whole, the oncologist here will declare victory because the tumor may have gone from this size down to this size. But there's a residue of cells that remains behind the cancer stem cells, and the reason why that's so bad is that obviously they can survive and weeks or months later regenerate an entirely new tumor. That, in fact, is one of the explanations of the source of much of the clinical resistance one sees in the oncology clinic, where it's been very difficult to eradicate the tumor. But until the last 5 years, we haven't understood the problem. And this is one of the major explanations, not the only one, of the problem. That tumors, solid tumors, are actually heterogeneous. Majority populations that may be more sensitive to therapy, and minority populations which not only are more resistant, but have the inherent potential of regenerating entirely new tumors, because remember, these are by definition tumor-initiating cells. In this case, they may reinitiate the new tumor at the site where the previous tumor resided, or maybe elsewhere in the body. And here's some evidence of this. Here, somebody looked at 8 different or 10 different cancer lines. These lines are over here are more epithelial. And these lines, which are more epithelial, they are sensitive to chemotherapeutic. You see here, these 4 channels to the left. These 5 channels over here are more mesenchymal. And these cells, which are more mesenchymal and therefore more stem-like, are resistant to therapy. It's a day or night difference. The epithelial cells with this fingerprint are killed off by the therapeutic. The mesenchymal stem cells with this fingerprint here, quite different, are resistant to killing by a cancer stem cell. Or here's another way of representing this. Here's a whole bunch of different cancer cell lines. And when I say cancer cell line, I mean tumor cells that are grown in the petri dish in an incubator. And here is the extent to which a chemotherapeutic drug will kill these cells. So here you have a lot of growth inhibition. Here there's a lot of killing. Down here, there's hardly any killing. The epithelial cells over here in the left half, they all are much more sensitive to killing. Up to 80% of these cells are killed by a particular drug. And here you can see there's extensive killing of these cells. The mesenchymal cells, the stem cells over here, they're much less responsive. In other words, they're killed to a much lesser extent by this standard chemotherapeutic drug. And that begins to solidify what I said before. Mesenchymal cells, mesenchymal cancer stem cells, are much more resistant to chemotherapeutic killing. In showing you these graphs, I'm only trying to reinforce what I was saying right here, that the effects of treatment are to leave behind these intrinsically more therapy-resistant cancer cells. So, 2 people in my lab, Tamar Under and Piyush Gupta, thought up the following strategy. Could they look for drugs that specifically kill cancer stem cells? Because I've just told you that in general, the available drugs don't do very much against cancer stem cells. The cancer stem cells have some kind of inherent resistance. And what they did was they produced essentially unlimited numbers of cancer stem cells by forcing these cells through the EMT. If you put everything, what I've just said together, you realize that if you can experimentally force a non-cancer stem cell through an EMT, you can create essentially unlimited numbers of cancer stem cells. You can make them by the millions or the billions. And then you can look for drugs that specifically kill the cancer stem cells, but leave behind and don't kill the non-stem cells. And so this is the work of Piyush Gupta. He started in my lab. He did much of it in the laboratory of Eric Lander next door after he left my lab and became a postdoc there. And as you heard, he's recently been hired back here, not because we like to feather our own nest, but because he landed on top of a job search with several hundred other competitors. And next door, the Broad has these robotic systems which allow one to look through hundreds of different drugs, thousands of drugs, for those that can particularly kill cancer stem cells. So the plan is, first of all, you have a robot introduce cancer stem cells in each one of these wells. I don't know how well you can see them. This is something like a 384 well plate here. Each one of these wells has cancer stem cells in them. And with that arrangement, then what you do is you do what's called high throughput screening. Specifically, you put into these wells different drugs and ask the robot to tell you whether the drug put in here or the drug put in here or the drug put in there has been successful in killing off the cells that you inserted there, the cancer stem cells, which I've just told you before you can make in essentially unlimited numbers. So they undertook, Piyush Gupta undertook essentially a high throughput screening. 16,000 compounds that they happen to have, a library of these compounds as it's called. And they asked which ones of these actually could preferentially kill off the cancer stem cells. So again, in some of these wells they put cancer stem cells. In other wells they put non-stem cells. Well, this is a mistake here. This should be non-stem cells in other wells. And then they had the robot inject all these compounds into the wells, and the readout was which of these compounds could preferentially kill the cancer stem cells rather than the non-stem cells. And here is the results of Piyush Gupta's screening. Here we'll look at these two graphs, and this is this is the pharmacology of drug development. And what we're seeing here is on the horizontal axis, the abscissa, these are higher drug concentrations. And over here in the vertical, this is the amount of cells surviving. So here all the cells survive, here they all get killed. Here are some non-stem cells in this blue curve right here. These are non-cancer stem cells, and the more drug you give them, the more of them are killed. In the same experiment, one can ask the following: how do cancer stem cells respond to this same drug? This is, by the way, a drug that's used routinely in the oncology clinic. And what you see, the red line, is the cancer stem cells. And they behave differently. These two lines are obviously displaced one from the other. But more importantly, at a drug concentration that kills off, has killed off the non-stem cells, the cancer stem cells have survived. They're not yet killed off. All the non-cancer stem cells have been killed off at this drug concentration, but the stem cells have not been killed. The same is here for this drug. It's like Taxol. The non-stem cells are killed off here at this drug concentration. The cancer stem cells, they tend to survive quite well, the same treatment, and are killed only at a much higher drug concentration. And this validates, once again, what I showed you before, what I argued before, which is to say, just to jump back to this, that the cancer stem cells are more resistant to killing by many standard available agents. So what happened next? I'm glad I asked that question. Okay, so they discovered out of these 16,000 compounds, about 20 which actually preferentially do the exact opposite. They kill the cancer stem cells more effectively than the non-stem cells. And they worked up two. One of them is salinomycin and the other is abamectin. And these two are antiparasitics, it turns out. Why they showed up here, no one has any idea. Salinomycin is used in chicken feed to keep away parasites in chicken coops. So the next time you go to KFC, you may actually be imbibing a small amount of salinomycin. It's up to you. Okay, so here what we see is salinomycin, which is actually a rather non-toxic drug. It kills the red cancer stem cells at a lower level of drug than it kills the non-stem cells. Here you see the curves are reversed. Here the cancer stem cells are killed more readily than the non-stem cells, and it's true for this. Here the displacement is only a factor of 2. Here this drug is killing the cancer stem cells about 6 or 8-fold more effectively. But it's exactly the opposite of what I showed you before with the behavior of conventional drugs. And here we see the effect of that. Here's a population of cancer cells. Here's the non-stem cells. Here's the stem cells. You treat them with Taxol, which, as you may know, is used very frequently in the clinic, and then you look at who survives the Taxol treatment, the standard chemotherapy treatment. And among the survivors now, 70% of the survivors are cancer stem cells. They started out at 5, now they're 70. Not good, because those are the bad cells in terms of recurrence. If you look at salinomycin, which you can get at KFC, then, oh, I didn't say that. I might, who knows if somebody, they'll get back to me with some anti-defamation libel suit. Anyhow, you look at salinomycin, after the treatment, the cancer stem cells have almost been wiped out. There's hardly any of them left. And this validates the general notion that one can preferentially, selectively kill cancer stem cells with certain agents. We have no idea why it works this way, but the important take-home lesson is that one can devise means using this strategy of generating large numbers of cancer stem cells. One can devise search strategies to identify compounds that selectively and specifically kill the cancer stem cells rather than the non-stem cells. And that would, in principle, seem to be a very good thing. But, I say, is this the answer to cancer? And it's probably not even if one can make these compounds, even if one can kill off cancer stem cells, there may be a problem. Here's what we would anticipate would be the end result of getting rid of the cancer stem cells with the KFC compound, salinomycin. You get rid of the cancer stem cells, the tumor loses its source of self-renewal, and now the tumor as a whole melts away. And everybody can indeed declare victory. But over the last couple of years in my lab, we have discovered that this diagram I showed you before actually has to be revised. It's not the way I showed you before. In fact, the arrows that went in this direction also have to be, one arrow has to be added in that direction. Pretty interesting idea. From non-stem cells, you can make new stem cells. And we can discuss how that happens, but there are a variety of situations where it happens. And this holds important implications for eliciting durable clinical responses to therapy, because here's the implication. Here I just showed you this is the desired scenario, but what if this happens instead? You get rid of the cancer stem cells, and now what happens is the surviving non-stem cells regenerate new cancer stem cells, which I think will happen. And that has an important conceptual implication in terms of being able to generate new types of durable anti-cancer therapy. In the future, when we wish to treat tumors and get a durable clinical response, we'll need to use conventional agents to get rid of the non-stem cells and novel agents, whatever they be, to get rid of the cancer stem cells here. We'll need to get rid of both populations. If you would have asked me 5 years ago, how are you going to cure solid tumors? I would have said, well, I have no idea. There's many problems, but I certainly would not have factored into my logic the whole existence of these cancer stem cells, which represent a very important and critical source of therapeutic resistance and clinical relapse. So here are the people that make all this happening. I'm just sitting on my hands most of the day. They do the work. These are the young people whom you are supporting. Not me, it's they who are doing the work, they who have the original ideas. They come up with the energy and the creativity and thinking out of the box that makes all this possible. And to the extent that I one day have to retire, which must happen sooner or later, it's them whom I will miss. Of course, I'll miss you too, David, but it's them whom I'll really miss. Thank you for coming. questions, yes. We have allotted some time for, and I actually finished pretty much on time.
I follow what you said, but I got like a chicken and egg question.
But let's take KFC out of this, though. We don't mention them again.
At the start, how does that first cell become cancerous? And how does it work? Is it like it becomes one of these tumor-generating cells?
Yes.
Or is it a regular cell that went bad? You know,
So
good stem cells that could have gone bad.
So the question is, how does the tumor start? And that's another whole hour. And if you're patient, and everybody else is, sure. Easy for you to say. They're hungry. All right, anyhow. The fact of the matter is, in order for a normal human cell to become a cancer cell, it has to go through a series of 4, 5, 6 changes to become a frank cancer cell. It doesn't happen from one to the other. And that process is likely to involve stem cells and non-stem cells, but we don't, although there's some nice speculations how that might happen, that doesn't really map on what I've told you today. Obviously, cancer stem cells will play a role in that. It's probably the case that the way a tumor actually develops is a normal stem cell becomes a slightly abnormal stem cell, becomes a more abnormal stem cell, and you have a whole progression of stem cells which progressively become more and more abnormal and ultimately, frankly, neoplastic. That's the rough answer to your question, the hand-waving answer, which is all I'm capable of today.
Bob, if this is a strategy of double one-two punch, would you, knowing what you know today, would you need less of the first drug, or would be less toxic because you don't need, you know that you're actually acting on two different...
Yes, so the question is, if one can develop a cancer stem cell agent, will that reduce the need to have high doses of, let's say, very toxic standard chemotherapy? And that's a possibility, but we don't know at present. It's just a possibility. At this stage, we're years away from putting things into the clinic. This is not something that's about to be introduced into humans, because these drugs need to be optimized and their toxicities need to be tested, and then their efficacy in killing tumors carried in mice needs to be tested and so forth. So that's 5 years, 3 years down the road. Sorry? You'll see me there, God willing. Okay.
Were there any candidates in the drug screening that were effective against both the stem cells and the non-stem cells?
So this is a very interesting question. I always tell my student, when somebody asks a particularly good question, never compliment that person on having asked a good question, because if you don't compliment everybody else later, they're going to feel like dirt. But nonetheless, that was a good question. I didn't call. Yours was actually outstanding. In fact, so obviously outstanding, I didn't think it warranted mention, anyhow. Well, we digress. Anyhow, it could be there are certain compounds, certain chemicals that can hit both the stem cells and the non-stem cells. That would be wonderful. But importantly for our discussion today, we need to keep in our vision, on our radar screen, the fact that there are these two different populations which need to be analyzed separately, because if we don't get rid of both of them, we will not have the hope of durable clinical response. It could be that certain agents, for reasons we don't understand, actually attack both of them with equal efficacy. The bulk of conventional chemotherapeutics, however, don't kill the cancer stem cells very effectively. Sir?
Yeah. When you're undertaking these tests, how do you choose 1,000 or so candidates out of the endless universe of possible compounds?
Well, what happened was that 16,000 compounds in the high throughput screen were just taken off the shelf. The Broad now has 300,000 compounds which were put through the same thing. And one does really a non-biased screen, asking which ones of these 16,000 compounds kill the cancer stem cells much more than the non-stem cells, without any preconception as to their identities. And that's all done by robots. Sometimes our postdocs and graduate students think of themselves as being robots, but these are real robots. And the robots, and that in bioinformatics identified, let's say, 22 compounds, which were selectively killing the cancer stem cells out of those 16,000. Of those 22, 4 or 5 were actually available in amounts, in physical amounts, that allowed further experimentation to happen. So seleninomycin was partly rationally discovered and identified, and it was worked up in part because there was a lot of it around. Sir, in the back?
Is there any risks of the cancer stem cells creating in the lab through forced EMT behave or react from real cancer stem cells?
Yes, definitely. So the question is, do the cancer stem cells that we make as laboratory models misrepresent the biology of real cancer stem cells? And the answer is definitively yes, which means that as this work is going on, one is at the same time looking at spontaneously arising cancer stem cells from human tumors to make sure that we aren't being deluded and deceived by the peculiar idiosyncratic behaviors of lab-generated cancer stem cells. That has to happen very quickly. Otherwise, we can go down some kind of rose garden path thinking we've done something interesting when in fact it turns out to be just, in effect, an artifact. In the way back?
There seems to be systems homeostasis to keep the populations at a certain percentage. So that when you get rid of the CLCs, it unleashes the mesenchymal potential.
Yes.
How are they communicating, and why are the CLCs suppressing the other populations?
Well, excuse me, Hilo, that's also a very good question. We don't understand what's going on. I understand your question, but we don't understand how the stem cells and the non-stem cells intercommunicate. In other words, the stem cells must sense how many non-stem cells there are so that one maintains a proper balance in the other direction as well. And that kind of dynamic equilibrium is something that is beyond our knowledge at present. But I think we'll figure that one out over the next 3 or 4 years. That's not unknowable. It's just unknown at present.
What period of time is involved in what you've been showing us, and what does that say about the rest of the time you need?
I'm glad you asked that question. I had a freshman biology teacher who said, "I'm glad you asked that question," and then rephrased the question to a form that he could actually answer. Now, a better question would be, in that the fact is, it could well be that what we now know could eventually end up in a drug that could be tried in phase 1 clinical trials, let's say in 3 or 4 years. Is that what you're asking? That could be the time scale, which is a bit more rapid than has traditionally been the case. Not 10 or 20 years, but 3 or 4 years, because these days, these technological innovations like the robotic machinery and bioinformatics, along with advances in our biological understanding of, let's say, stem cells, have greatly accelerated the pace with which one can move from the laboratory bench to phase 1 trials in the clinic.
Why is it so hard to think or guess to make progress against cancers like pancreatic cancer? And is there anything you think should be done that could speed up that?
So the question is, why is it so difficult to make progress against pancreatic cancer? What do I do now? They're all good questions, and we're all children of the Lord. Okay, so pancreatic cancer is an interesting case. Why are pancreatic cancers so lethal? We understand the genes that are mutated in them, but I haven't the vaguest idea why they've proven so refractory to treatment. And you'll say, well, why is that? And the fact of the matter is there are fundamentally, critically important issues in tumor biology which we just don't understand today. We haven't learned all the answers yet. And obviously, we need to learn the answer to that question. Why are pancreatic cancers so refractory to treatment? And I cannot give you a coherent answer, and I would argue no one else can at present either. We have some clues and some speculations, but the answers are not in. There are some people who think that we know all we need to know now about the basic biology of cancer. If we just applied this information, we'd have all kinds of new drugs and treatments. But it ain't true. There are big questions that remain unanswered. There are people who say to us, well, if you cancer researchers would just talk to one another more and collaborate with one another more, then everything would be solved. I spend half of my time in the airplane talking with cancer researchers all over the world. It's not as if we're hiding our light under a bushel and not talking with one another. And it's not as if we don't collaborate with one another, sometimes promiscuously. That's not the reason. The reason is that there's some ideas we haven't grasped yet about why, for example, pancreatic cancer, which is a very challenging tumor, eludes any kind of treatment to date that people have devised. Sir?
Is there something inherent in your research methodology using drugs as a therapy that suggests that gene therapy wouldn't work or would be tried later, or is it not a question?
Well, can I compliment him? Okay. Hilo says it's okay. Very good question. The fact of the matter is, gene therapy has been proposed as a way of treating cancers. And the notion there is to put genes into cancer cells which would reverse their bad behavior. That's the essential outline of the idea. And it's a great idea. Conceptually, it's wonderful. It has one major problem, and it's a fatal problem. We don't really know how to insert genes into the billions of cancer cells that coexist within a tumor. We don't know how to get them there. It's a delivery problem. And that delivery problem was unsolved 10 years ago, and it remains largely, if not entirely, unsolved today. So there's many great ideas, but that one cannot be materialized, it cannot be realized, I should say, because of the problem of not knowing how to get genes into tumors. We know how to get chemicals into tumors because chemicals are very small and can slither through all the interstices of the body's tissues. But genes are big things, and we really don't know how to deliver them. Sir?
You said you don't have a problem in communicating your research with others.
Correct.
And then you showed that terrific picture describing how much more quickly the robotic use in high throughput analysis has made things go faster.
Yes.
Is there a way of communicating, not like robotics, but that could be more efficient, communicating research, findings, associations than you have now?
I'll tell you two things. First of all, the Broad next door puts on a public website whenever it makes one of these discoveries within several months of the finding. In other words, it's not as if years intervene before the cancer research community has access to those findings, which I think is to be applauded. Secondly, the single most important invention in modern biology is something that's never discussed, and that's a website called PubMed. PubMed is a website in which there are now, I think, 18 million scientific research publications that you can access literally within seconds. The person who's done that was astounded when I told him he made more of a difference to the progress of modern biomedical research than anyone else besides maybe Watson and Crick. And the fact of the matter is, if I want to find out something now, I don't have to make a telephone call. I don't have to write a letter. I don't have to ask people's advice. I just get on my laptop and I put in a couple search terms, and within seconds or a minute, I can find out lots of what's happened in this area and been published up to the last couple of weeks. And so one should cite that not as an indication of how good cancer researchers are or how smart we are, but how the pace of communication has enormously accelerated the pace of research.
I meant without having to put your hands on the keyboard and your eyes on the screen.
Yes.
And that work be done robotically so that your work is associated with others and you get some filter.
The answer is no. We don't understand how the human brain works, but the fact is we're dealing here with very complex conceptual structures which robots are decades away from being able to deal with. The human brain is still irreplaceable. We can flatter ourselves to saying, and I suspect it will be for decades. That's why we're talking here together. You didn't come here with the knowledge that you could learn everything you've just heard today by going to some website. You came because you thought, I believe correctly, that by hearing it come out of the horse's mouth and being here and thinking about things as I was talking about them, you could assimilate something rather than just getting in on a conference call or going to some website. And so the ergonomics of the human brain and the human intellect are such that you can't robotize interactions like these. And that's why students go to class too. Sitting in front of the professor, for better or worse, is important to them because it helps them to figure out what's going on and to wrestle with these issues in ways that we have no idea how the human mind really works.
Isn't there a very tough tension between the collaborative approach and the sharing of information and the ownership of intellectual property, which David earlier told us is an important revenue source, or could be revenue source for Whitehead?
There is in principle a tension there. And the tension we resolve by the following. Before we publish something, we don't delay the publication, but we take out applications to make intellectual property patent rights. Provisional. What you know more about this, I sense, than I do. The fact of the matter is we do not delay our publication to the outside world in order to begin to exploit it commercially. We apply for intellectual property and we publish at the same time. And that way both needs are met.
Who owns the intellectual property?
Who owns the intellectual property? Why don't you answer that, since I just work here?
If intellectual property is generated at Whitehead Institute, it's owned by Whitehead Institute. So it's, you have to think about, in some cases there are provisions that involve funding agencies. But for instance, NIH basically grants those intellectual property rights to the research institution.
And you share it with the principal investigator?
Yes. Well, the royalties, right. So the invention, I mean, I don't think we will want to get into a long discussion of the of the IP flow, but Bob will, together with his co-inventors, file for IP protection, and that protection is sought and prosecuted by Whitehead Institute. And of course, it may be, we talked about collaboration before, we're collaborating with many other institutions. There may be joint IP, which is shared with other institutions. And then at the end of the day, should there be a royalty stream that's generated, a healthy chunk of that is shared back with the inventors. Yeah.
Madame.
Stepping back maybe miles, could you describe the role of creativity in the intellectual development of these brilliant young students of yours, and perhaps identify a common thread in their prior education?
That's a little bit of a tough one. I can only tell you about how I run my lab. And that is, I breed in the people who work in my lab a healthy disrespect for the principal investigator. I tell them that I'm excited if they go off on tangents here and then, without telling me for months thereafter. I do that with the knowledge that in my own lab, many of the innovations, the great majority of them, to be truthful, over the last 3 decades have not come from my head, but from the heads of these young people. Now, I haven't really touched on what happens inside the human brain when one is creative, but the fact of the matter is the reason why we talk to one another so often is that we're constantly trying to make connections between different areas of research. In my lab, we meet every day of the week for at least an hour to talk about a new research journal, to critique each other's work, to have an open discussion about some brainstorm somebody had, just to create a cauldron, a fermenting cauldron where people are throwing ideas around and all of a sudden having insights of where this is really connected with that. So that would be the best way I could describe the creativity. I'm sure somebody who's interested in psychodynamics might do it in a more effective way.
How many of them remain in this country?
I'm sorry?
What
percent remain in this country? Well, it's interesting. People often say, well, why are we allowing training all these foreigners in our country when they're going to go back to their home country and develop competitive industries there, or competitive academic institutions? The fact of the matter is, if we did not have a continuing, unceasing inflow of young and bright people from across the globe into this country over the last century, even to this day, we would be a third-rate scientific power, third-rate. The fact of the matter is we need those people because they drive our research, they lead our research, and the great majority of them stay in this country. Without them, we would be lost. If you think the average American kid who goes to high school in this country, who spends college years going to tailgate parties at football games, is filling our is staffing our laboratories, it ain't so. It's a tragedy, but American education, native-born Americans, whatever that means, has failed to address our need of continually injecting new, bright, young, ambitious, hardworking people into our research apparatus, and keeping them here after they've trained with us. Over the last 5 years, for the first time, some of the Chinese postdocs have actually started going back to China. So one of the dangers we now have is if there's an implosion of scientific funding in this country, we will, on the one hand, no longer be able to attract foreigners to work in our labs to the extent we have over the last half century. And two, after they're trained, they may go back to their other country. But keep in mind, by the way, when I talk about the cancer research community, I'm not talking about the American cancer research community. I'm talking about the international community. Between now and since September, I've been to Europe 3 times talking with various cancer research communities there about various aspects of what we're doing. So it's an interwoven web, and we're not in competition with them. We draw from their brilliant successes, and they draw from ours. So we have to keep that in mind. The chairman of the board, you were so sequestered back here.
First of all, thank you, and you don't have to comment on the quality of this question.
Already, it's a very good start.
You have to stand so close.
Well, it's closer you stand, the more threatening.
Well, I couldn't resist a comment. I appreciated the clear answer about who owns the intellectual property. I'd only note that intellectual property has been probably the greatest growth area of legal work and lawyers that we have going.
Remember what Shakespeare said.
No, but I have to respond to that. He only said that because the lawyers were the protection of right and honesty in those years. That's why they want to kill them. This is really true.
I didn't hear.
My question is, on the 16,000 of the compounds and 300,000, it was interesting that you mentioned one that was effective happened to deal with parasites. And
yes.
I thought that seemed interesting. So that's one question. The other is, can you draw from what you find with these many thousand compounds and go down into a more directed, instead of having to just kind of really shoot all around? And the parasites was also kind of an interesting side issue.
So we have no idea why 2 antiparasitics turned up in this screen. The strategy in general is to find 20 or 30 or 40 good drug compounds, see what they have in common, pick ones that have the greatest potency and selectivity, and then, as they say in the pharmacy industry, derivatize them. Add different chemical groups to the left side of them, to the right side of them, up and down, and make variant forms of those molecules and see whether, in fact, those variant forms are more potent. And in fact, in this salinomycin drug, one has started to do that and find far more potent agents than the one that was initially described in the screen. So there's actually a logical and rational advance, but I can't tell you because I don't know why antiparasitics showed up in this screen. It's a mystery.
Well, to piggyback on his question, what does that say about the origin of those cancers? We've got many different types of cancers.
Sure. So what does discovering antiparasitics tell us about the origin of the cancers? It says the following. About 1.5 billion years ago, nucleated cells were invented. About 600 million years ago, animals were invented. And once animals were invented, a whole circuitry was used for all animals on the planet and even for the parasites. And therefore, many of the components that are present in the parasites and essential for their existence are also present in our own cells. And therefore, there's some underlying commonalities. And to the extent that we will one day understand why that antiparasitic from KFC works the way it does, it will come down to the fact that there is a common biochemical circuitry in the parasite and in human cancer cells which somehow is affected equally by that agent. But having said that, I haven't told you what that pathway is. That's far beyond us still.
So I think I'm going to thank Bob for this fine presentation.